Imagine taking a perfectly safe allergy pill, right? Like one you've taken a dozen times before. And then you sit down to a perfectly normal breakfast where you drink a glass of grapefruit juice.
Just a standard morning routine.
Exactly, just a normal morning. But then, a few hours later, that combination triggers a lethal heart arrhythmia. No warning, no overdose, just a catastrophic, invisible collision right in your bloodstream.
It's terrifying to think about.
It really is. So today we are looking at the highly dangerous and, well, sometimes brilliantly engineered world of drug interactions, or DDIs.
Yeah, it really shatters the illusion of medical precision, doesn't it? Because we like to think of medicine as binary. You have a localized problem, you take a pill, and it just fixes the problem.
Right, like a mechanic swapping a part.
Exactly. But the human body isn't a simple machine. It is a chaotic, flowing ecosystem.
Okay, let's unpack this, because if you are listening to this, you've probably seen those tiny warning stickers on your prescription bottles. But what's actually happening underneath?
A lot more than we usually realize.
Right, so we've pulled from medical history, pharmacology textbooks, and some really wild clinical case studies to uncover the hidden mechanics of what happens when medications cross paths.
And we're going to explore why these collisions happen.
Yeah, the accidental discoveries that completely changed modern medicine and, how doctors were actually hacking these very interactions to save lives. But jumping into the history here, I was actually surprised to see that the whole concept of drugs interacting with each other is relatively new.
It is surprisingly recent, yeah. If you look at medical history before, say, the 1950s, doctors were mostly prescribing one drug at a time.
Oh, really? Just one?
Pretty much. So the modern concept of a drug interaction didn't really exist because the biological opportunity for them to collide simply wasn't there.
That makes sense.
It only emerged when synthetic polypharmacy, meaning, the standard medical practice of taking multiple synthetic medications simultaneously, when that became the norm.
So by inventing more drugs and prescribing them all at once, we basically invented the biological traffic jam.
Precisely. We built the cars and the roads, and the crashes inevitably followed. And that traffic jam forced clinical pharmacology to mature into an entirely distinct discipline.
Because they had to figure out what was going wrong.
Exactly. Like in the 1960s, physicians started noticing these terrifying patterns. For example, a patient would be on the blood thinner warfarin, right? And their blood work was perfectly stable. Then they'd be given a simple sulfonamide antibiotic for an infection. Their warfarin dose never changed, but suddenly they started bleeding unpredictably.
Wow. Their clotting time just spiked out of nowhere.
Out of nowhere.
I read that at the time they knew the antibiotic was somehow secretly amplifying the blood thinner, but they had absolutely no idea why. Like they were flying blind.
They were totally flying blind. They just knew that a drug considered safe in isolation could become incredibly dangerous purely because of what else was swimming around in the patient's system.
Right.
So to understand how they collide, we first have to understand the biological highway they travel on.
Which brings us to the liver, the engine of it all. I know there was a massive breakthrough in the early 1960s regarding how the liver actually processes these chemicals.
Yes, the great chemical processing plant. Between 1962 and 1964, biochemists Ryo Sato, Martin Klingenberg, and Sunio Omura identified and characterized this specific superfamily of oxidizing enzymes in the liver.
And this is the famous one, right?
Yeah, it became known as the cytochrome P450 family, or CYP450.
CYP450, it sounds like a droid from Star Wars, but it is arguably the most important mechanism in your body that you've probably never heard of.
That is an understatement. Discovering the CYP450 superfamily finally gave pharmacologists the mechanistic why.
So it explained the underlying biological process.
Exactly. It explained how one drug could mysteriously change another drug's blood levels by altering how it is oxidized and cleared from the body.
Oh, wow.
And once they had that biological anchor, a clinical pharmacist named Philip D. Hanston realized they could map this all out. So in 1971, he published a landmark book called Drug Interactions.
So instead of doctors just having a chaotic, scattered list of case reports saying, like, don't mix drug A with drug B, Hanston actually organized the chaos into a system. How did he break it down?
He essentially split all of this into two massive clinical buckets: pharmacokinetic and pharmacodynamic interactions.
Okay, pharmacokinetic and pharmacodynamic.
Right. And that textbook division is still the core of pharmacology over 50 years later. Pharmacokinetic interactions are all about logistics.
Meaning how the drug moves around?
Yeah. It's when one drug changes how much of the other drug gets into the body, where it distributes, or how fast it's cleared out. Most of the time, this is happening at the metabolism step, right there in the liver with those CYP450 enzymes.
I want to try an analogy here to sort of visualize those pharmacokinetic logistics. Think of it like a crowded nightclub.
Okay, I like where this is going.
So if CYP450 is the bouncer standing at the back door, letting drugs out of the club so they can be cleared from your body, an interaction is when one drug goes up and completely distracts the bouncer. Because the bouncer is busy, the other drug is trapped inside the club. The music keeps playing, the drug keeps partying, the concentration builds and builds, and eventually things get highly toxic.
That is a remarkably accurate way to picture clearance rates. I love the bouncer analogy.
Thanks.
But the other bucket, Hansen-identified pharmacodynamic interactions, is completely different. That is when the blood levels of the drugs are perfectly normal.
So the bouncer is doing his job perfectly.
Exactly. The clearance is fine. But the two drugs are altering the actual effect at the biological target. They are pushing on the exact same physiological lever in your brain or your organs.
Right. Well, before we look at the levers, I want to stick with that first bucket, the bouncers, because something in the research didn't totally make sense to me.
Oh, what was that?
If we have known about these specific liver enzymes since the 1960s, and we know exactly how this biological nightclub works, why is the severity of an interaction still so hotly debated?
Oh, yeah, that is a huge issue.
Because if you look at your own medicine cabinet and cross-check it online, databases like the FDA, Micromedex, and Lexicomp, they frequently disagree. One system will flag a combination as a major risk, and another will just say moderate. Why are they still arguing over the math?
Well, it really comes down to the messy reality of clinical evidence. We might know the precise enzyme involved, but the evidence proving the real-world danger varies wildly.
How so? Like, the testing is different.
Yeah, sometimes the FDA is looking at rigorous, randomized, crossover pharmacokinetic trials with hundreds of patients, clearly showing a lethal spike in blood levels.
Okay, so that's hard proof.
Right. But other times, a database is flagging a severe warning based on a single, isolated case report published in a medical journal 20 years ago.
Oh, wow. So just one person had a bad reaction once.
Exactly. The definition of an interaction has had to drift over time. It used to mean any measurable change in the body. Now it's shifting toward a change large enough to plausibly alter a patient's actual clinical health, and finding that exact line is incredibly difficult.
Okay, so let's look at what happens when that line is definitively crossed. If we go back to the bouncer analogy, there's a mechanism called enzyme inhibition. Basically, when the bouncer isn't just distracted, but gets entirely knocked out cold.
Yes. And CYP3A4 is the star player here.
CYP3A4.
Right. It's one specific, highly active enzyme in that P450 family, and it is responsible for metabolizing roughly half of all clinically used drugs. Half.
That's insane. Just one enzyme handles half of our medications?
Just one. So if a second drug comes in and chemically blocks CYP3A4, the first drug's concentration climbs to toxic levels, even though the patient took their exact prescribed dose.
Which brings up the terfenadine story from the 1980s. Most people knew it as Seldane.
Oh, Seldane, a classic example.
Right, it was a massive blockbuster allergy drug because, unlike the older stuff, it didn't cause drowsiness. But I was reading about why it worked, and it's fascinating. The pill you actually swallowed wasn't the active drug, right? It was something called a prodrug.
Correct. Terfenadine is a prodrug, meaning the parent compound itself is nearly inert in terms of curing your allergies.
So it does nothing on its own.
Basically nothing. It relied entirely on your liver, specifically that CYP3A4 enzyme, to metabolize it, breaking it down into the active form that actually gave you relief.
But the catch was that the unmetabolized version, the parent compound, was highly toxic to the heart's electrical system.
Highly toxic. Under normal, everyday circumstances, your liver metabolizes the prodrug so incredibly fast that the toxic parent drug never has a chance to build up in your blood.
So the bouncer kicks it right out.
Exactly. But if a patient took Seldane alongside a potent CYP3A4 inhibitor, like erythromycin, which is just a common antibiotic, the enzyme gets chemically blocked.
And the bouncer is knocked out.
Yeah, the toxic parent drug builds up, prolongs the heart's electrical reset time, and triggers a chaotic, lethal arrhythmia called torsades de pointes.
All because you took a standard antibiotic with your standard allergy pill. I mean, it's terrifying. It was completely lethal for some patients, and the FDA had to pull Seldane off the U.S. market entirely in 1998.
It was a massive wake-up call for the industry.
Yeah, and they eventually replaced it with Allegra, which is literally just the safe, already active metabolite of celdane, completely bypassing the liver enzyme problem entirely.
Which was a very elegant solution.
It was, but here is the part that genuinely shocked me. It wasn't just other prescription pills knocking out that enzyme. It was breakfast.
Ah, yes. The infamous grapefruit juice accident of 1989.
I just love the sheer serendipity of this story. So a Canadian researcher named David Bailey is running a study to see if alcohol interacts with a blood pressure drug called felodipine.
Right.
But to make sure the control group didn't realize they were drinking pure alcohol, he needed something to mask the incredibly strong taste. So he went with grapefruit juice.
And what he discovered was pure accidental science. What's fascinating here is that when they looked at the blood work for the juice-only control group, the felodipine levels were just off the charts.
Way higher than expected.
Vastly higher. It turned out that grapefruit juice itself acts as a massive enzyme inhibitor in the intestines. But the biological mechanism of how it does this is what's truly remarkable. It's something called suicide inhibition.
Suicide inhibition, how does that work?
Well, the juice contains these chemical compounds called furocoumarins. When they encounter the CYP3A4 enzyme, they trick it.
They trick the enzyme.
Yeah. The enzyme tries to metabolize the furanocoumarin, but the chemical structure essentially acts like superglue. It permanently binds to the enzyme's active site, irrevocably gumming up the machinery.
Oh, wow.
It doesn't just distract the enzyme, it irreversibly destroys its ability to function.
So a single glass of juice completely wrecks the system. How long does it take to recover from that?
The inhibition can last anywhere from 24 to 72 hours from a single glass.
3 days.
Up to 3 days, yeah. Because the enzyme is permanently destroyed, your body literally has to wait for your DNA to synthesize and manufacture entirely new proteins to replace the enzymes that were lost.
That is wild. I mean, that completely shatters the misconception that grapefruit is just some minor kitchen table curiosity. It's a massive medical concern.
Absolutely.
If you're listening to this and you take statins for cholesterol and you have half a grapefruit with your morning coffee, you are massively spiking your blood levels for days because you're actively destroying the mechanism that clears the drug.
And we know how devastating spiking cholesterol medications can be. Consider the Bakol disaster in 2001.
Oh, I've heard of Bakol.
Yeah. The cholesterol drug cerevastatin, known commercially as Bakol, was often prescribed alongside another lipid-lowering drug called gemfibrozil. This combination inhibited the clearance of Bakol so severely that the drug built up and caused patients' muscle tissue to literally start breaking down.
Right, I've heard of that condition, rhabdomyolysis.
Yeah.
But how does muscle breakdown actually turn fatal?
When the muscle fibers break down, they release a protein called myoglobin into the bloodstream. Now, your kidneys are responsible for filtering your blood, but myoglobin is a large, sticky protein.
So it clogs the filters.
Exactly. It clogs the delicate filtration tubes in the kidneys, destroying them. It caused fatal kidney failure in dozens of patients, and Bakol had to be pulled worldwide.
That's tragic.
It really is. It proved definitively that an interaction alone can kill a drug. Bakol worked, but the sheer mechanics of its clearance made it too dangerous to exist in a world where it might bump into gemfibrozil.
So up to this point, we've focused heavily on the liver. We've seen the absolute chaos when blood levels spike because the liver enzymes get blocked. But we need to look at Hanston's second bucket, pharmacodynamic interactions.
Yes, the shared levers.
Right, because what if two drugs never meet in the liver at all? They don't share a bouncer, their metabolic pathways are completely different, and their blood levels stay perfectly normal.
Bringing this up is crucial because it dispels a very common, dangerous misconception. People often assume, well, if these two drugs are processed by entirely different organs or pathways, they can't possibly interact.
But they absolutely can.
They absolutely can. If they push on the same physiological lever, the end result is a profound interaction.
So give us an example of pushing on the same lever.
The most consequential modern example is the combination of opioids and benzodiazepines like Valium or Xanax.
Okay.
Both of these classes of drugs independently suppress the brainstem's respiratory drive. The brainstem is the primitive part of your brain that involuntarily tells your lungs to expand and contract.
But they act on completely different receptors in the brain, right? They aren't competing for the same molecular parking spot.
Correct. No metabolism or direct chemical interference is involved. But if you take them together, the depressant effect is additive.
Meaning they stack.
Exactly. The blood levels of both drugs could be perfectly within the normal, safe, prescribed range. Yet together, their combined weight on those different receptors completely turns off the brain's signal to breathe.
Oh, wow.
It is a lethal synergy, and it's a massive driver of the modern public health crisis, which is exactly why the FDA mandated severe boxed warnings for that combination in 2016.
I was actually reading about a case from 1984 that perfectly illustrates this shared target danger, the Libby Zion case.
Yes, a very famous case.
It's famous because it actually changed the laws of how doctors operate. I know she was an 18-year-old girl admitted to a hospital in New York, and she was already taking an antidepressant called phenelzine, which is an MAOI.
Right.
And an overworked resident gave her a painkiller called meperidine. But I couldn't wrap my head around how those two specific drugs caused a total systemic failure.
It's a textbook, tragic pharmacodynamic interaction. To understand the mechanism, you have to look at serotonin. Her antidepressant, the MAOI, works by intentionally stopping the brain from breaking down serotonin, keeping levels elevated to improve her mood.
Okay, so her brain already has a high baseline of serotonin that isn't being cleared out.
Exactly. Then, the resident administered meperidine. Meperidine, as a side effect, triggers a massive acute release of serotonin from the nerve cells.
Oh, I see.
Yeah, so you have one drug dumping a massive flood of serotonin into the synapses, and the other drug completely blocking the brain's ability to sweep it away. The synapses get overwhelmed.
Which leads to serotonin syndrome.
Right. Serotonin syndrome is a cascading state of neurological and cardiovascular overactivation. Her temperature spiked dangerously, she went into cardiac arrest, and tragically passed away.
And the investigation didn't just stop at the drugs, did it?
No, it looked at the systemic failure of the hospital. The resident who prescribed the painkiller was working incredibly long, unsupervised, sleep-deprived shifts.
So they were exhausted.
Beyond exhausted. The subsequent investigation determined that fatigue played a major role in missing that fatal interaction. That specific physiological collision directly led to the 1989 New York regulations that legally limited medical resident work hours, which eventually forced a fundamental change in medical training across the entire country.
So a drug interaction didn't just change medicine, it changed the law. Honestly, this whole deep dive makes pharmacology sound like a terrifying minefield. You eat a grapefruit, and your heart medication spikes. You take a painkiller with an antidepressant, and it causes a fatal neurological flood. Are we just at the mercy of these collisions, or have drug developers figured out how to use this biological machinery to our advantage?
That is the brilliant flip side of the coin. Pharmacologists aren't just reacting to these collisions, they are actively engineering them to make therapies better.
Here's where it gets really interesting. Hacking the system.
Precisely. Take Parkinson's disease, for example. The primary treatment relies on a drug called levodopa, which the brain desperately needs to synthesize dopamine. But there's a problem. If you just swallow a levodopa pill, enzymes in your peripheral body will chew it up and destroy it long before it ever crosses the blood-brain barrier.
Doesn't even make it to the brain.
Exactly. So pharmacologists combined it with a second drug called carbidopa.
I was reading about this. Carbidopa essentially acts as a chemical decoy, right? Like a bodyguard.
A perfect description. Carbidopa sacrifices itself. It purposely binds to and blocks those peripheral enzymes, taking the hit so the levodopa is ignored and can safely travel up into the brain.
That's incredibly clever.
It is. Because of this engineered interaction, doctors can use a drastically lower dose of levodopa, which prevents the severe nausea and cardiovascular side effects that used to plague Parkinson's patients.
Wow. And I saw another engineered interaction in the sources that I think almost everyone listening has probably experienced, Augmentin.
Oh, definitely.
Anyone who has had a stubborn sinus infection or an earache has probably been prescribed Augmentin. It's amoxicillin, which is the antibiotic, combined with a chemical called clavulanic acid. The clavulanic acid doesn't kill the bacteria at all. Its only job is to run interference and disable the bacteria's defense enzymes, so the amoxicillin can do the actual killing. It's like a microscopic tag team.
It's elegant chemistry. And perhaps the most life-altering example is in modern HIV therapy. To keep the HIV virus suppressed, patients need a steady, constantly high level of antiretroviral drugs in their blood.
So the levels can't drop.
Right. If the liver clears the drug too fast, the blood levels dip and the virus aggressively rebounds. So doctors routinely prescribe a companion drug called ritonavir, but they prescribe it at a dose so incredibly low that it has zero therapeutic effect on the HIV itself.
Wait, so why add another pill to the regimen if it doesn't fight the virus?
Purely to act as a booster. Ritonavir happens to be a potent, deliberate inhibitor of CYP3A4. It is prescribed solely to knock out the cancer at the liver's back door.
Oh, I get it.
Huh.
By keeping CYP3A4 completely occupied and blocked, the primary HIV drug stays trapped in the patient's bloodstream much, much longer. Which means the patient doesn't have to wake up and take a handful of pills every 4 hours just to maintain those blood levels. They can take a smaller dose, far less frequently.
Exactly. And the beautiful irony here is profound. The exact same biological mechanism, CYP3/4 inhibition, that accidentally killed patients taking the allergy drug Seldane, is now actively and deliberately utilized as a tool to make HIV treatment highly effective, convenient, and life-saving.
That is incredible. We are literally weaponizing the traffic jam.
Oh, we really are.
But, this leads me to the math problem of modern polypharmacy. Because if we know all these rules and we understand the enzymes and we have sophisticated computers tracking all our pharmacy prescriptions, why are DDIs still one of the most persistent leading sources of medical harm? I mean, if a patient is on 10 medications, they don't just have 10 units of risk, right?
Right, and that is the most dangerous misunderstanding of polypharmacy. The risk doesn't grow linearly, it grows combinatorially.
Combinatorial.
Yeah. Interactions happen between pairs of drugs. If you are taking 2 drugs, that's one possible pairing. But if you are taking 10 medications, you don't have 10 risks. You have 45 possible 2-drug pairings.
45. That is a massive leap.
Every time you add a single new prescription to a patient's regimen, the web of possible collisions explodes exponentially. And this isn't just about synthetic prescription drugs. There is a pervasive, stubborn myth that natural inherently means safe.
Oh, yes. I know exactly where you were going with this. St. John's wort.
It is the perfect counterexample. St. John's wort is a very common over-the-counter herbal supplement that people take for mild depression or mood. But pharmacologically, it is one of the most potent known inducers of CYP3A4.
Inducer, meaning the exact opposite of an inhibitor.
Correct.
Earlier, I tried to picture it as giving the bouncer an energy drink, but that's not biologically accurate, is it? It's not making the existing enzymes work faster.
No, it's actually altering genetic expression. The compounds in St. John's wort travel to the liver and trigger your DNA to synthesize and manufacture a massive amount of new CYP3A4 enzymes.
Okay, so a better analogy is that it calls the security company and hires 50 extra bouncers for the nightclub. The doors are thrown wide open, and the club is emptied out in minutes.
Exactly. It ramps up the liver's clearance capacity dramatically. So consider a patient who recently had a kidney or heart transplant.
Okay.
They take St. John's wort because the whole ordeal has left them feeling down. The herbal supplement triggers the liver to build an army of new enzymes. Those enzymes rapidly metabolize and clear out their life-saving anti-rejection medications like cyclosporine way too fast.
Oh, the drug levels just drop completely.
The protective drug levels plummet, and their immune system wakes up and rejects the transplanted organ, all from taking a natural herbal supplement from the grocery store.
Wow. But if I am an organ transplant patient and I go to the hospital, why doesn't the electronic health software just scream SDOOP when a dangerous pair is logged?
The tragic reality is that it does scream. The problem is it screams entirely too much.
Really?
Yeah. This is known in the medical community as the signal versus noise debate, leading to a phenomenon called alert fatigue. Remember how we discussed that the definitions of interaction severity vary widely?
Right, the FDA versus Micromedex argument.
Because hospital electronic health record systems are designed by corporations terrified of being sued for missing anything, their algorithms are calibrated to be overly sensitive. They flag almost everything.
So the doctor or pharmacist is just getting an endless barrage of pop-ups on their screen.
A relentless barrage. Studies show that busy physicians override well over 90% of lower severity interaction warnings.
90%, that's almost all of them.
Exactly. They literally learn the muscle memory to ignore them because in their specific clinical context, the vast majority of these alerts are medically trivial.
That makes sense, but it sounds dangerous.
It is. The counterintuitive twist here is devastating. By making safety information more available, we actually made clinicians so completely desensitized to the alarms that they end up accidentally clicking past the rare fatal alert. Over-alerting causes the very harm it was designed to prevent.
Wow. More warnings do not automatically mean better safety.
Not at all. And if we connect this to the bigger picture, you can see how relevant this balancing act still is today. Think back to the COVID-19 pandemic and the rollout of the antiviral pill Paxlovid. Paxlovid is actually a combination of two drugs, and one of them is ritonavir.
Wait, the same ritonavir booster they use for HIV therapy?
The exact same one. It is used in Paxlovid deliberately to inhibit CYP3A4, intentionally knocking out the bouncer so the actual antiviral stays in your system long enough to fight the COVID infection.
But because it severely disables that vital liver enzyme, Paxlovid has a massive, terrifying list of dangerous interactions with everyday medications.
Yes. From blood thinners to statins, pharmacists were suddenly thrust into extreme vigilance, manually cross-checking massive medication lists during a global crisis where speed was critical, all while battling that software alert fatigue.
That sounds incredibly stressful. So what does this all mean? Let's recap. When drugs collide, they essentially fight on two battlegrounds. They're either battling over logistics and clearance rates, pharmacokinetics, where liver enzymes like CYP450 act as the bouncer, or they're battling over shared targets, pharmacodynamics, where two drugs pull the exact same physiological lever in the brain or body, like the tragic Libby's iron case.
Perfectly summarized.
We've seen how these interactions can pull blockbuster drugs off the market, like Seldane or Bakil. But we've also seen how brilliant pharmacologists can engineer these collisions to save lives, like with Parkinson's or HIV. And crucially, we've learned that slapping a warning label or an endless stream of software pop-ups on everything doesn't actually solve the human error problem.
Right. It is a profoundly complex system, and ignoring the underlying biological nuance is dangerous.
And if you are listening to this, this is exactly why your personal health literacy matters more than ever. With modern polypharmacy, you might get a prescription from a cardiologist, another from a psychiatrist, and grab an herbal supplement like St. John's wort from the grocery store.
And they might not be talking to each other.
Exactly. There often isn't one single doctor seeing the whole board. Understanding the mechanics of how these chemicals interact in your specific biological system is your best defense.
And that idea of your specific biological system brings up one final critical reality.
Oh.
We've talked all about how drugs interact with these liver enzymes, but what happens if the genetic blueprint for your enzymes is fundamentally different from mine?
Different how?
There is a rapidly expanding field called pharmacogenomics. What if, due to your unique personal genetic code, you entirely lack the gene required to physically translate and build a functional version of an enzyme like CYP2D6?
So the bouncer just doesn't exist. He was never even born.
Precisely. In that case, a standard safe dose of a perfectly normal medication could be incredibly toxic for you while doing absolutely nothing to the person sitting right next to you.
That is mind-blowing.
And this isn't some distant science fiction future. If you go to a modern psychiatrist today for certain severe antidepressants or an oncologist for cancer treatments, they might swab your cheek first to read your genetic code and map your unique liver enzymes before ever writing a script.
So they are actively preventing these deadly drug collisions before they even happen.
Yes. The era of the standard dose is already dying.
It brings us right back to where we started, doesn't it? That dream of pure medical precision. No more muddy waters, no more guessing if the biological nightclub is crowded. Just a clean, visible map of exactly how the chemicals will behave the second they enter your bloodstream.